Okay, I'm recording this morning, and my plan for today is to have lecture stuff and announcements first, and then at the end of class we'll go over exams, because there's still here and there a few people that still need to make up exams, and that way, like, the people that can go over the exam can, and the people that shouldn't yet shouldn't. One important announcement is that today for lab, and basically every week for lab, we will start at 1:45 p.m. and we'll be in Klein 121. So that's going to be our big room location. And what I plan is that each one of these lab weeks, the first 45 minutes or so, I will talk about how to do the next steps of your paper. And then the best use of the remaining time is to kind of work on it while we're all in there. And I can consult with you. I know that you have something at 2:45, is that right? How about you?
I'm making a genetics test.
Today?
Yes.
I will be recording these sessions. So we'll be starting each Thursday at 1:45 in Klein. I will be broadcasting those on Teams. And I will be broadcasting those on Teams, and you would just use the same Teams link as everything else in this class. And I probably also will record them, but they won't, I don't know, they may be pretty useful. I'll probably do a screen share. Remember, if you have not turned in your article choices, 2 article choices, those are due like right now. But if you haven't turned them in, go ahead and turn them in as soon as you can, because I want to look through those and let you know if I have, let you know what my suggestion is to continue with. I'm probably not going to get those looked through before 1:45 today, but the sooner you turn them in, the sooner I will let you know which one I would suggest. Any questions about lab stuff? What time are you starting your makeup?
Right around 2.
Yeah, don't do your makeup exam. Okay. So we're going to continue with the topic of how do proteins move to their correct cellular location. And we had stopped last time on vesicles, talking about vesicles and how vesicles form in the first place, and that requires these 3 proteins called clathrin, adaptin, and dynamin, which are here. This seems too big today. Try this again. Oh, it's getting so big like that. Bear with me. Just when I think I've got all the technology things down and figured out and working properly. Something like this. Oh, it's just starting right there. Strange. We'll just do this method. That'll work. Okay, so when a vesicle is forming, remember it's going to have cargo that it's carrying as its reason for existence, let's say. So the cargo has to be attaching to a particular receptor, and that binding of cargo to receptor, that would use the 4 forces, and it's non-covalent. But that binding event is going to then recruit adaptin to the cytosolic side of this receptor, and that combo then recruits clathrin. And the cooperative assembly of these clathrin proteins helps pull the vesicle out of its originating membrane. When the clathrin has almost coated the whole vesicle, there's a protein called dynamin that comes in at the end and uses ATP energy to kind of pinch that vesicle off and get it finished. Then the vesicle technically is free, and it will briefly still have clathrin and adaptin on it, but not long. They disassociate, and the vesicle is now ready for transport. I think we made this note last time, but if in case, this vesicle is then going to get associated with motor proteins that will carry it to its next location. So they don't actually just kind of drift around in the cell on their own. They have to be carried by a motor protein. And I think week after next, we talk about the carrying, moving of vesicles process. Okay, so the vesicle comes from a membrane, and the vesicle is going to head to another membrane carried by a motor protein. But when it gets to its destination or target membrane, this is depicting where the vesicle is supposed to end up and the membrane it's going to fuse with. That's the target membrane. And just like having the process of pulling a vesicle out of its originating membrane requires energy, the process of fusing a vesicle with its target membrane also requires some energy. So what needs to happen here is that the vesicle membrane has to merge and fuse with the target membrane. So there's a system of proteins that help this process when the vesicle gets close enough to the target membrane to do this fusion process. And it's kind of like if you've been on a boat out on the lake, you have to drive the boat kind of close to the dock, and then you turn the engines off, and then there's usually like a rope throwing process sometimes, and that last little bit is a slow pull of the boat into the dock. This is kind of a similar system. So we see this vesicle up at the top. I guess I can't reach it so much. It's got its cargo, which looks like a brown pear. That's the cargo. It's got the receptor. And it also has a couple of other proteins that have been attached to this vesicle. One of them is called V-snare. V-snare. And the V in this V-snare terminology stands for vesicle. And that V-snare is an integral membrane protein that's really firmly wedged into the vesicle membrane. And it's long and basically helical, and it's kind of waving around on the exterior. It's not rigid like this, it kind of flops around. And The other protein that is on the outer surface of the vesicle is called RAB. The snare and the RAB terminology do not totally match with the function. So if an acronym ever totally matches with the function, I teach you what it means. If not, it's because the RAB, whatever it stands for, it is not obvious how it relates to this. But the RAB protein has binding affinity with a protein from the target membrane called a tethering protein. So these two can bind together through the four forces non-covalently. And that interaction is kind of like the throwing of the rope, if we could compare this to boats. The tethering protein is really long. It also waves around. It has flexibility to kind of catch this RAB protein. And when these two bind, this tethering protein changes shape to get the vesicle close. Get it close. And then the V snare protein will do its function. So the V snare always has a partner called T snare, and T stands for target. So we have several versions of these, but they occur in matching pairs, or let's say complementary pairs. So there are particular V snares that match or have complementary binding to particular T snares. So regular plasma membrane has a certain kind of T snare that matches with a V snare targeted protein on a vesicle that's supposed to go to the plasma membrane. There would be separate pairs for lysosome and separate pairs for Golgi, let's say. And when these two helical type proteins get close enough together, they twist and they wrap around each other like so. And that twisting, wrapping around, will pull the vesicle close enough to the target membrane that it mushes the phospholipids together, and then they fuse. And once it starts to fuse, then this part goes quickly and the cargo comes off. Let's say something about the cargo coming off. So whatever this compartment is, and right now it's extracellular, but it could be the interior of a lysosome, or it could be inside the Golgi or whatever. Whatever this compartment is, its conditions cause the cargo to dissociate. So whereas in the interior of the vesicle, the cargo stays stuck to the receptor, once the vesicle fuses and its liquids mix with the liquids of this compartment, the conditions of this other liquid are different in salt concentration, maybe pH, different electrolytes. It's a different environment, and this environment is not enough to keep the four forces together. So this cargo is going to dissociate. But you need RAB interacting with the tethering protein to get kind of close, and then you need a matching complementary twist binding of the V snare and the T snare to get the vesicle close enough to actually fuse. To me, this concept is cool, but it's overrepresented on like standardized tests of cell biology. that they really like to use these terminology. And if you just remember that these have to do with vesicle fusion, that's helpful. V snare, T snare, vesicle, and target membranes. This is a picture of how the twisting process occurs. So they've taken out the RAB protein and the tethering protein. They're still there, they're just not shown. And when those get the V snare and the T snare close enough that they can flop around and interact, then it goes really fast. They twist, twist, twist around each other and pull the vesicle. It is cool. Okay. So we're kind of going backward in time now to talk about the beginning of proteins going into the endomembrane system. So let me go back and show where we are in the bigger picture of endomembrane traffic. So remember that anything that is going to be targeted to outside the cell, or Golgi, or endosomes, or lysosomes, it's got to go into the endoplasmic reticulum first, and it's going to be translated through a pore into the ER. So we're going back to this step right now, and talk about some clinical type implications. So the protein that's going into the ER, whether it's soluble or whether it was supposed to be in the membrane, it's still got to have the correct fold. It's got to have the correct 3D shape so that it will do its correct function wherever it's going. If the proteins are getting into the ER and they are misfolding, they're making the wrong shape, cells are able to sense that, and they will produce these chaperone proteins that are folding helpers. So this red protein here that they're calling chaperone, that is a folding assistant, let's say. And if folding is not going properly, the cell can make chaperone proteins and send them into the ER to help try to fix the problem. If the chaperone protein is able to assist the folding properly, then this cargo can get in a vesicle and proceed to its destination. But you need to have quality control here. This is a quality control before the cell spends so much energy to send a wrong protein onward. Make sure it's folding correctly before it leaves. If chaperones are needed, this is kind of how the cell senses and how it produces chaperones. This is not a key figure, but I really like this concept because it relates back to a particular disease, which is diabetes. So I'm kind of gonna spend some time here, even though this is not a big test concept, because it's clinically interesting. So it starts with, there's misfolded proteins, and inside the ER there are some receptors that can detect misfolded proteins. So this is kind of this binding is a warning signal. Oh my gosh, we need chaperones in here to help folding. So the detection of the misfolded protein activates a transcription factor that travels into the nucleus through the nuclear pore complex, and it will turn on transcription of chaperone genes. So the gene will get transcribed, the messenger RNA will go through, it'll get the 5' cap, poly A tail, splicing if it needs splicing. Then it will proceed to leave the nucleus and go out on a ribosome, start getting translated. It has a signal. It would have that N-terminal signal sequence that this is a protein that's supposed to go into the ER. The ribosome translates through the channel, the chaperone protein folds, and then it tries to help. It tries to help properly fold this messed up protein. So this picture, I also like this because this is showing like the life history of the chaperone protein in a very condensed form. It has to be transcribed, it has to be translated, it has to be translocated, and then it will do its function. Mm-hmm.
So what initially causes so many proteins to misfold?
It's not that many proteins, fortunately. And why they misfold a lot of times is they've got this protein also had a separate gene it became transcribed from. And it could be because it's got a point mutation at a critical spot that makes the early steps of folding not go well. But why they misfold, it's different for different proteins. And it's fortunately not all the time, because that would be maladaptive. When you have class, is Plant where Juan's been talking about maladaptive? He was telling me the other day he had been talking about the word maladaptive, and I don't know what class it was from, but if this persisted down through generations of organisms,
I think that was a seminar.
Seminar?
Someone's presentation was talking about maladaptation or whatever.
Really?
I think so.
Last year?
Oh, last year? No.
This year?
This year, yeah, like last week.
I think somebody did say maladaptive. I think it was Liz White, because did she just say maladaptive? Anyway, if you have class with him, that's one of his favorite things to talk about. And it is cool, but if there were a protein that consistently through generations folded wrong, it would be maladaptive and it would be selected against, basically. But why they fold wrong, it's different for different proteins. If all of this happens, and this is expensive and time-consuming for the cell to try to fix this problem, but if the chaperone cannot fix the problem and the protein is staying like this, it's kind of building up in the ER and crowding out the normal functions of ER, and the cells can sense that too. And if they sense that that's the problem, then they will undergo cell death. Just like this cell is hopeless. Just I can't even just undergo programmed cell death. And this, oh my gosh, I just took that slide out. That's unfortunate. This can happen with insulin being the misfolded protein, or insulin receptors being the misfolded protein. It's insulin. It's insulin that happens the most. And write this down. What cells of your body are making insulin, or where? Yeah, in the pancreas. So if this is happening in the pancreas, and insulin, so in the pancreatic beta cells, if this is happening and insulin is constantly misfolding, that can lead to death of pancreatic beta cells. and the person stops having the ability to make insulin, and that can lead to diabetes. So people that were working on diabetes research actually figured out a lot of this info about chaperones and ER folding, and this basic knowledge came out of diabetes research, which is cool. Another very important thing that happens in the ER is the attachment of sugars, or what we would call covalent modification of the part of the protein that is lumen side or eventually would be extracellular. The main place where proteins would need sugar chains is if they are heading out to the plasma membrane. The sugars tend to be extracellular. So the addition of the sugars is done in the ER, and the addition of disulfide bonds, like if this had a disulfide bond needed because it's going out to the harsh conditions of extracellular, that would be done inside the ER as well. And adding disulfide bonds kind of oxidizing conditions. You need oxidizing conditions to do that. Just like you need reducing conditions to break a disulfide bond, you need oxidizing conditions to make them. And oxidizing conditions are kind of too harsh for the cytoplasm, too harsh for the cytosol, because that's a more intense chemistry. So the cell kind of contains that in the ER, and that's where disulfide bridges form. And remember that all of this side of this protein will end up being extracellular or in the lumen of part of the endomembrane system. And say that this was going into the lysosome eventually, that's super acidic. That's a time when you would need disulfide bridges to hold the protein together in acidic conditions. You don't really need disulfide bridges in the cytosol, because the cytosol is like happy preschool man. Everything's easy, nothing's harsh, all kind and living. You need the disulfide bridges when you're going into harsh conditions. So uncontrolled, sometimes uncontrolled conditions. So everything that has come in these three slides, the folding, quality control, the assistance of the chaperones, the adding on of sugars and disulfide bonds, those are endoplasmic reticulum functions. The next steps are going to take place in the Golgi, or the Golgi apparatus, if we want to be old school. And the Golgi is responsible for basically figuring out and tagging vesicles so that they end up at their correct destination. So the ER can make proteins and get them into vesicles. The Golgi is responsible for getting the vesicle formed with a particular cargo, getting its V snare on, getting its rab on, and sending it on its way out to other parts of the cell. So it's doing some modification, but also sorting of proteins into their correct vacuole. Notice here, all of this is Golgi, and any separate part is a vesicle that's coming off. But the Golgi has a polarity to it called the cis side and the trans side. Have you learned this before? Okay, great. I bet Dr. Vaughn teaches it. Yeah, so how do you know what cis and trans?
Cis is closer to the nucleus.
Yeah, and trans is more distant. So like the word cis, it kind of means the same. Trans is across, so cis is on the same side, trans is further away. And for those of you that didn't have Bio 1 with Dr. Vaughn, you could go back and label this. This is the cis Golgi on this side. This is the trans side. And so vesicles are generally going to be moving from the cis direction toward the trans direction, but they transport between layers of the Golgi. So there can be a vesicle moving here to here, further processing the contents here, move here to here, and eventually the long distance vesicles will come out the trans side of the Golgi. When I first came to King and taught cell, the students had not heard of cis and trans Golgi, so it's helpful. I could have just gone to this picture. So once a vesicle leaves the trans Golgi, it should have been filled up with a fairly specific cargo, and have its RAB and have its V-snare and have a particular destination. So we could draw on here that this vesicle, it's got RAB, it's got its V-snare, it's got its cargo, and remember the cargo is on a receptor. And this vesicle heading outward is supposed to be dumping its contents outside the cell, and we call that a secretory vesicle. So what's in it is just different for different cells and different vesicles, but it could be this could be a lot of different things. This could be anything that the cell is secreting. So it could be insulin, it could be mucin, thank you, mucin that combines with water to form mucus. It could be neurotransmitter, if this is a neuron. The neurotransmitter vesicles tend to kind of wait out here until it's time for them to fuse. But things like insulin and mucin have sort of a more regular, like, keep fusing, dumping the contents. And notice here, if the vesicle waits before fusion, if it has regulated fusion, that's going to come from a signal outside the cell that's like hormone, neurotransmitter, dump vesicle now. Do they not have? They don't. Oh, that's the top part. Yeah. So the other ways that vesicles can dump their contents is in a constitutive pathway. So I actually need to change mucin. Mucin would be constitutive. Insulin is some constitutive and some regulated as well. But you could write mucin up here or just draw arrow. That's something that the cell needs to export all the time, like on an hourly basis. So stomach acid, basically constitutive. Mucus, constitutive. What else? Sebum, constitutive. All of that's traveling in vesicles. And notice up here too, there's something else in this vesicle that's important, which is new membrane lipids just for upkeep. When the vesicle fuses, it's bringing new membrane lipids out to the plasma membrane just as a refresh, a constant supply. So every cell has to be sending some vesicles out to the plasma membrane, if only to replenish the lipids. And when they fuse, it's exocytosis. Here's an awesome picture of vesicles fusing that are cram-packed full of insulin. So this dark featureless blob is almost to the level of crystallized insulin. It's so concentrated and packed together. So the insulin would then be traveling into the bloodstream, circulate around the body. And there should be grabs and V-snares hanging off this vesicle. So the last content we have in this section is to talk about the inbound vesicles, or the inbound pathway to come into endomembrane system. So that would be the green arrows here, inbound traffic. And the most extreme example of inbound traffic of materials into a cell is phagocytosis. This picture on the right is one of my top 10 favorites. So both of these are showing phagocytosis, which not all cells can do. But in our bodies, we have white blood cells as part of the immune system that can engulf particles that they consider dangerous. So on this side, we've got a white blood cell that is engulfing 2 rod-shaped bacteria. And on this side, we've got a white blood cell that is engulfing 2 at the same time. This is so awesome. 2 different yeast cells. Like, this is a tough job. It's taking on 2 yeast cells, and they are all almost together half the volume of this cell. So this is the most extreme type of endocytosis. And phagocytosis in this way, there's no vesicle coming in. It's a crawling or an extension. It's an extension of the cell around an object. It's not making a vesicle big enough to pull this in. The cell crawls around it. It flows around the bacteria, or it flows around the yeast, and then it makes a compartment. So this is going to happen until the object is surrounded in a new compartment. And that new compartment is an example of one kind of early endosome. So you can imagine, once these cell extensions finish crawling around these bacteria, both bacteria are going to be enclosed in a membrane, And that essentially has become a new compartment that is two bacteria surrounded by membrane, technically in the cytoplasm of this cell. So that's the capturing part. And once an object is captured, it can be considered an early endosome. And early endosomes, with the right signals, can turn into a lysosome. This is so cool. Like, I'm going to surround you, and then I'm going to dump acid into you and digest you and use your parts to build more me. It's so cool. And white blood cells, this white blood cell can make it work to destroy those yeast, or it dies trying and takes the yeast with it. That's the real extreme kind of endocytosis. That's the most exciting. There are other less exciting. There are other less exciting, but are important, and that are done by more cells that can't do phagocytosis. So it's pretty important for cells to be able to pick up cholesterol. They need to pick up cholesterol. And this particular cell, it has receptors on the outside facing extracellular that bind LDL. Which is low density lipoprotein. Those LDLs are proteins plus cholesterol. Proteins plus cholesterol in a blob. And cholesterol is so fatty that it can't circulate in the body without proteins to help it dissolve. So we call this LDL, and this is the form of what people call bad cholesterol. There's another version called HDL, which is good cholesterol. So in this process, the cells are using receptors to bind LDL and initiate inbound vesicle traffic. So clathrin, adaptin, dynamin still apply. This would still be moved by motor proteins. It comes in and fuses with an endosome, and the endosome can sort. It can detect, this is something that the cell needs to go further, so send it to lysosome, get the cholesterol away from the protein, and the receptor gets sent back out to the cell to pick up more. The endosome is able to sort. A lot of cells would also do this whole process of bringing in some stuff from the outside into an endosome non-specifically. Why would they do that? That's because cells sample their external environment to monitor the conditions. Monitor the conditions out there and get information. Like if we were trapped in this room and we didn't have windows, we wanted to know what's going on outside. It would be like, let's gather some of the environment and bring it in here so that we can look, see what's happening. That's non-specific endocytosis. And if you've heard of penocytosis, peno comes from the word drink. Where the cell is just getting a little bit of the outside fluid, bring it in, check it out, see what's there. That's penocytosis. You heard that word before? Good. I remember it like there's a kind of wine called Pinot with a T. So I remember it. I don't like wine that much. I'll drink it if it's free, but don't buy it. But that's how I remember that, the drink the environment. I talked about those in the wrong order. Same slide. Okay, so remember that the destination, one of the common destinations of materials from outside that go through endocytosis, join an endosome, if they're going to continue to be inbound, they're going to end up in a lysosome. In some ways, it's not, there's two ways that materials can end up in a lysosome. Either the endosome sends the materials to the lysosome, or sometimes the endosome becomes a lysosome. So if you think about the giant engulfment, phagocytosis, this cannot be sent to a lysosome. It's just too big. But the compartment that it ends up in can become a lysosome around it. And that's also pretty cool. But in the end, whether the lysosome is pre-existing or whether it forms out of an endosome, it contains a set of vicious enzymes, every kind of hydrolase basically that exists, because the function of a lysosome is to dismantle whatever is inside it. We're going to start with this. So a lysosome would contain nucleases to break down DNA, RNA, proteases to break down proteins. What would a glycosidase do?
Break down sugar.
Break down sugar. These are tearing off sugars that were on proteins. And so I just asked that to get you used to thinking about, I don't know what that word is, but it's sugar ACE. It's tearing off the sugars that were stuck on the surface of proteins. Break down lipids, take off phosphates, take off sulfates, and dismantle phospholipids. So whatever the cell gets in a lysosome, the enzymes are in the lysosome to do the breakdown process. All of these types of enzymes are optimized to work at an acidic pH. They are optimized to function at pH 5, which is lysosome acidity level. If the lysosome happens to break open and these enzymes join the regular cytoplasm, and they're like unleashed on the cell, they will pretty quickly stop functioning out here because the pH is wrong. So pH 7 versus pH 5, how much more acidic is pH 5 than 7? About 100 times more acidic. Remember that pH is a log scale. So this is 100 times more acidic than the cytosol. That acidity level is required for these to work and maintain their function. So if they happen to burst out, they would do some damage, but the pH would quickly inactivate them out here. One really important component of lysozymes is that they have proton pumps. They maintain this pH by spending ATP to push protons into here. So they have a particular proton pump that hydrolyzes ATP to power pushing protons into the interior here. They couldn't function without that. And then our last slide today is showing the concept of if a particle is so large that it can't travel into they can't fit in a vesicle, its compartment can basically become a lysosome by fusion with endosomes that are already acidic. So if you want to make this giant compartment into a lysosome, the best way to do that is to fuse endosomes that have acid and have the hydrolytic enzymes with this, and it would be multiple of these, not just one, but multiple, coming over and joining this new compartment, start digesting that, and then the contents of a lysosome get recycled. So let's draw that onto the previous slide. It's kind of important. Do it here. So this set of vicious enzymes, whatever their hydrolyzed products, they're going to be sent out from the lysosome, and the cell will use those to build more cell. So those are going to be used by the cell as building blocks. This is not the main way that cells get nutrition. This is a way that cells either recycle internal proteins or destroy dangers from the outside. But the cell might as well use the products from those and not let it go to waste. So they do use those products. And notice here at the top we've got a danger, something dangerous that was engulfed by the cell. But down here, this is a mitochondrion. This is a worn out mitochondrion. So cells will also use this endosome fusion destructive process to recycle the contents of worn out or sick organelles, so to speak. And that's called autophagy. Autophagy versus phagocytosis. Autophagy. Let's have our break right now, and I'm going to stop recording. Probably also stop broadcasting on Teams. But remember that if you're on Teams, we will meet at 1:45 today in Klein. And I also will be running Teams during that session. So we'll see you at that time.